Welcome back to the Deep Dive. Today, we're really getting into the weeds of cell biology. We're tackling a major hurdle in respiratory research. We have this fascinating study that looks at a really clever workaround, the air-liquid interface, or ALI culture, using A549 lung cells. Our goal here is to figure out how scientists used, well, basically physics, just changing the cell's environment, to sort of nudge a common cancer cell line towards acting like healthy human lung tissue, at least partially.
It's a great example of innovation when, the ideal biological material just isn't available or practical.
Exactly. And to really get why this ALI approach is so neat, you first need to understand why studying the lung at the cellular level is just so darn difficult. We're talking about the alveoli, right? Those tiny air sacs deep in the lungs where all the oxygen and CO2 exchange happens. They're lined by mainly two kinds of epithelial cells. First, you've got alveolar type I cells, AECI, super thin, covering like 95% of the surface area. They're all about that gas exchange.
hmm And then you have the ones that cause all the headaches for researchers. Alveolar type 2 cells, AECII. These are more cuboidal, blocky cells. They're absolutely vital. They make surfactant, that crucial substance that keeps the air sacs from collapsing. And really importantly, a subset of these AECI cells act as progenitor cells, like stem cells for the alveoli. If the lung gets injured, they kick into gear, divide, and differentiate to replace damaged AECI cells.
Okay, so these ACII cells sound like the ones you'd really want to study, the key players.
They absolutely are. They're the gold standard. But here's the problem. Working with them in the lab is incredibly challenging. There are two big issues. First, just isolating enough of them from primary human lung tissue for, decent-sized experiments, extremely difficult. And second, this is the real kicker. When you do manage to isolate them and try to grow them using standard cell culture methods, where they're just submerged in liquid media in a dish,
Yeah.
they lose their identity. They pretty quickly stop making the things that make them AECI cells, like surfactant protein C. They just drift.
So the very act of culturing them traditionally destroys what you want to study. This study called that environment ineffective and artificial, which sounds about right.
Exactly. You have the right cell type, theoretically, but you can't get it to behave like itself outside the body. It's a major bottleneck.
Which is why researchers had to get creative and find an alternative.
Mm-hmm.
So the gold standard primary ACEI cells was basically off the table for routine work, and they needed to stand in. And they turned to a workhorse of lung research labs, the A549 cell line. It grows easily, reliably, but it's derived from lung cancer, specifically adenocarcinoma.
Right. It has that cancer cell advantage of proliferating indefinitely, but it's obviously not a normal cell.
So the big question became, could they take these cancer cells and, using some kind of special technique, push them towards differentiating, towards acting more like those crucial AECI cells? And that's where the air-liquid interface comes in.
That's the elegant part. If you think about the actual lung, those alveolar cells aren't swimming in fluid. They sit at an interface, right? There's a very thin layer of liquid on their surface, and then there's air in the alveolus. The ALI culture method is specifically designed in vitro, in the lab dish, to mimic that natural environment. The idea is that maybe the physical environment itself dictates how the cells behave.
Okay, so how does it actually work? Walk us through the setup described in this study. It sounds surprisingly simple.
It is, fundamentally. First, they take the A549 cells and seed them onto these special membranes. They used millicell inserts, which are like little porous cups that sit inside a larger well. The membranes were pre-coated with collagen to help the cells attach.
Right, giving them something to grow on.
Exactly. Then they let the cells grow submerged for about 24 hours just to establish a layer. And here's the key step. After that day, they simply remove the culture medium from the top, the apical side, inside the insert cup.
So the top surface of the cells is now exposed directly to the air in the incubator.
Precisely. But they're still getting nutrients because the bottom, the basal side, is bathed in the culture medium underneath the membrane. That's the air-liquid interface.
And they just kept them like that? For how long?
They cultured them for up to 3 weeks in this ALI condition, just changing the medium underneath every couple of days to keep them fed.
Okay, I'm still trying to wrap my head around this. You take cancer cells, put them on a filter, take away the liquid from the top, and that slight change in environment could actually make them start acting like normal lung tissue, override their cancerous nature, even partially.
Well, that was the hope. And the initial results, just looking at the cells physically, were pretty compelling, especially when you compare them to A549 cells grown the old way, submerged after 2 weeks. Those submerged cells, they looked pretty chaotic. Their researchers described them as having inerratic shapes, just Disorganized, irregular blobs, basically. Smooth surfaces, no real signs of forming proper tissue.
Okay, so the standard culture looked like, well, cultured cancer cells. What about the ALI ones?
The big difference, when they stained the ALI cultures using standard hematoxylin and eosin or H&E, you could immediately see they'd formed a proper, intact layer of epithelial cells. It looked organized.
And under the more powerful microscopes, SEM and TEM.
Even more dramatic changes. The scanning and transmission electron microscopy showed significant differentiation. The ALI cells were described as being more compacted, packed tightly together with clear cell junctions visible between them. Their top surfaces weren't smooth anymore. They were rough with abundant secretions, even signs of mucus. It looked much more like a functional barrier tissue.
Wow. And there was one specific finding from the transmission electron microscopy, the TEM, that really stood out. They saw a large number of lipid droplets and lysosomes in the cytoplasm. Why is that such a big deal? Like finding a smoking gun.
It really is, because those structures, lipid droplets and lysosomes, are considered classic ultrastructural hallmarks of type 2 alveolar cells, AECI cells.
What do they do?
Well, remember, AECI cells make surfactant. Surfactant is largely lipid-based. So those lipid droplets, they're likely storing the surfactant precursors or components, and lysosomes are involved in processing, packaging, and recycling cellular materials, including surfactant components via structures called lamellar bodies, which are derived from lysosomes.
I see.
So finding lots of these specific structures inside the cells tells you that the internal machinery needed for making and handling surfactant, a key ACII function, has been switched on, and it seems to be primarily because they were exposed to air.
That's fascinating. So the cells started to look the part, structurally, down to the tiny details inside. But looking right isn't the whole story. Did they actually start acting right, producing the right molecules? They needed molecular proof. What specific protein markers did they track to confirm this shift in function?
They focused on key markers for both types of alveolar cells, using techniques like immunofluorescence, immunoblotting, and quantitative RT-PCR to measure them. For type 2 cells, the main target was surfactant protein C, STC. They found it was moderately increased in the ALI cultures compared to the submerged ones. Not a massive jump, but definitely higher.
Okay, so some evidence for ACI function. What about type I markers?
This was actually quite significant. They looked at aquaporin 5, or AQP5, which is a water channel protein characteristic of the flat AECI cells. And AQP5 expression went up significantly in the ALI cultures.
Whoa, wait. A549 cells are thought to be derived from type 2-like cells, right? So the ALI environment is pushing them towards type I characteristics too.
It appears so, yes. It suggests the ALI condition doesn't just maintain or enhance the original phenotype, but might actually induce differentiation towards the other major alveolar cell type as well. It implies a level of plasticity.
Interesting. Were there other markers that supported this idea of forming a proper epithelial layer?
Definitely. They saw increases in thyroid transcription factor 1, TTF1. That's a really key regulator for lung cell identity. And they also found more mucin 5B, MUC5B, which ties back to those mucus-like secretions they saw physically.
Right. And you mentioned clear cell junctions earlier. Did they find molecular evidence for that too? That seems critical for barrier function.
Yes, and that's crucial. They looked at zonula occludens 1, ZO1. This is a major protein component of tight junctions, the seals between epithelial cells.
The glue that holds the barrier together.
Exactly. Finding increased ZO1 expression provides the molecular confirmation for those physical observations of compacted cells and clear junctions. It shows the cells are actively building a tight functional barrier, which is fundamental to how the lung epithelium works.
Okay, this sounds like a huge success then. They got the structure, they got the markers.
Yeah.
But you hinted earlier that timing was important. How long did this last?
Ah, yes, that's the catch. They tracked the expression of these markers over the 3 weeks of ALI culture. What they found was that the expression levels, STC, AQP5, ZO1, the works, were significantly boosted and peaked somewhere between 1 and 2 weeks.
Only 1 to 2 weeks.
Yes. They specifically identified that 7 to 14-day period as the best window for the alveolar property of this A549 ALI model. After that, the expression levels tended to decline.
That seems like a pretty significant limitation. On the one hand, you've coaxed cancer cells into behaving somewhat normally, which is amazing. But on the other, that normal-ish state is transient.
It absolutely is a limitation. It means the model is really well suited for studying things with a short time course, maybe acute lung injury, the immediate effects of a drug or a toxin, perhaps viral infection dynamics over a few days.
But not for chronic conditions.
Right. If you're interested in chronic diseases like fibrosis or COPD, or you want to see the long-term effects of a potential therapy that might take weeks or months, this model isn't ideal because the specific alveolar characteristics fade after that 2-week peak.
Okay, let's zoom out. We have this A549 ALI culture model. It manages to express some markers of both ACI and ACI cells, forms a decent barrier, but only really optimally for about 2 weeks. Why is this still considered so valuable? Is it just better than nothing?
It's much more than better than nothing. It's currently the most feasible and effective alternative for many types of studies. Like we said at the start, getting enough primary human AACI cells and keeping them functional in culture is incredibly difficult, bordering on infeasible for routine large-scale research right now. So this A549 ALI model, even with its flaws, provides a robust, reproducible system. It allows researchers to investigate aspects of alveolar physiology and pathology, how the barrier works, how cells respond to stimuli. That would be extremely difficult or impossible to do otherwise, especially compared to the clearly inadequate submerged cultures. It really fills a critical gap.
Got it. But we have to be clear-eyed about the downsides, the ones the researchers themselves pointed out. First and foremost, these are still cancer cells. A549s come from lung adenocarcinoma. They grow uncontrollably, which is useful in the lab, sure. But fundamentally, their ability to differentiate into normal tissue is inherently limited by their cancerous origin.
Exactly. That's the underlying issue that likely contributes to the other limitations.
Like the short duration, that 1 to 2 week functional window we keep mentioning, that severely limits the kinds of biological questions you can ask.
And there's another nuance related to the differentiation. While they saw those significant increases in the markers for type I cells like AQP5 and TTF1,
Yeah.
they specifically noted that they didn't actually see cells that looked like classic type I cells morphologically, those super flat, spread out cells covering the alveolar surface.
Okay. So the cells were making some type I parts, but they weren't fully remodeling into the final type I shape.
Precisely. It supports the conclusion that it's a valuable functional model, but it is still a partial mimicry. It's a pragmatic solution, a scientific achievement within certain constraints.
So if you're listening, the big takeaway here is pretty cool, I think. Faced with this really tough biological problem, recreating the lung's delicate air surface lining scientists didn't necessarily find a perfect cell replacement. Instead, they took a known, reliable, albeit cancerous cell line and used a change in the physical environment, that air-liquid interface, to essentially unlock a more specialized, albeit incomplete, function. It's a fantastic example of how sometimes the environment, the physics, can be just as important as the genetics in the lab.
It really is. And that leads to a kind of provocative thought, something for you to maybe mull over. Think about it. These A549 cells started as cancer, yet just by changing their physical surroundings, putting them at that air-liquid interface, they activated complex machinery to take on specialized roles, producing things normally associated with healthy AECI function. It really makes you wonder, in biological models generally, how much of what a cell does is rigidly fixed by its genetic code, its DNA blueprint, and how much is actually flexible, adaptable, maybe even forced or triggered purely by the physical and chemical context it finds itself in. Where's the balance between intrinsic programming and environmental instruction?
A really fundamental question for anyone trying to model life in the lab. Great point to end on. Thanks for joining us for this deep dive. We'll see you next time.